This video is going to overview the autonomic nervous system. So when we consider the human nervous system, it is the most complex structure in the known universe. We break it into parts. So there's a central nervous system, the brain and spinal cord. There's a peripheral nervous system, which includes nerves and ganglia outside the brain and spinal cord. Some of what the nervous system does is what's referred to as a sensory division, where it measures things like touch and taste and smell and sight and vision and relays that to the brain. And then, based on this information, the brain and spinal cord may decide to tell the body to do things. So there is a motor division. So the motor outputs, very often we think of, telling skeletal muscle to do things, especially voluntarily. And perhaps we think of this first because we're voluntarily aware of, consciously aware of telling, the body to run, et cetera. So that's what's called the somatic nervous system. And so the brain and spinal cord, when they give commands to the body, some of these commands go to skeletal muscle, and that's what we refer to as the somatic nervous system, or SNS. So here you can see in a cat, here's the spinal cord. Here you can see nerves going out to the muscles of the shoulder. And here you see skeletal muscle cells. So that's important. And students of anatomy, they then, study, it's, this nerve, the radial nerve that goes to the triceps brachii that tells it to extend the forearm, etc. However, there are more commands that go out other than those to skeletal muscle. So for example, the nervous system sends commands to cardiac muscle of the heart, smooth muscle, exocrine glands like sweat glands, endocrine glands, and there are even commands which go out to adipose and immune cells. And so therefore, this is a different part of the nervous system. Unlike the somatic nervous system, the SNS, where commands are sent to skeletal muscle, the autonomic nervous system, the ANS, sends commands to everything that isn't skeletal muscle: cardiac muscle, smooth muscle, exocrine glands, endocrine glands, and adipose. One other big difference we have to address first is if this is a cell of the SNS, there are dendrites, which collect electrical information related to the soma. Maybe the soma decides to go to an action potential at the trigger zone. And then this action potential spreads along the axon. The axon will then actually go to the muscle cell. So it is the neuron which leaves the spinal cord, which actually then goes through the radial nerve and reaches then the muscles of the triceps brachii. So this one neuron left the central nervous system. Its soma and dendrites were in the central nervous system, and yet, and then it arrives to touch the skeletal muscle. In the autonomic nervous system, that is not how it is structured. And so the autonomic nervous system not only has different targets, so once again, skeletal muscle for the SNS, and then kind of everything else for the autonomic nervous system: cardiac muscle, smooth muscle, exocrine glands, endocrine glands, adipose, immune cells. So the effectors are different, but also the structure is different because the cell which leaves the central nervous system is not the cell that arrives to give commands to the effectors. There are always two neurons, all right? Neuron number 1 and neuron number 2. Now that's not a very good set of names for them, so we'll rename them in just a second. So neuron number 2 has a soma and dendrites, but they aren't located in the central nervous system. So where do you put them? Well, we have a name for that. So if you have a cluster of soma inside, say the brain or the spinal cord, we call it a nucleus. So if you ask, here's the red nucleus, what would one find at the red nucleus? Or here's the nucleus of the oculomotor nerve, what would we find there? The answer is a cluster of soma. So inside the central nervous system, a cluster of soma is what we call a nucleus. If you have a cluster of soma outside the central nervous system, it's called a ganglion. So what is in a dorsal root ganglion? It's a cluster of soma, but it's outside the spinal cord. All right, so the second neuron has its soma and dendrites outside the central nervous system, so it's located in what's called a ganglion. This ganglion then helps us to name these neurons, because neuron number 1 is perhaps more accurately and descriptively called the preganglionic neuron. So the preganglionic neuron comes before the ganglion. It has its soma and dendrites in the central nervous system, and its axon travels to the ganglion where the synapse is. And then neuron number 2 is more descriptively called the postganglionic neuron. Its soma and dendrites are in the ganglion, but then its axon proceeds from the ganglion to the effector tissue. So a difference between the SNS and the ANS is that in the SNS, you have a single neuron leave the central nervous system and go to the effector, while in the autonomic nervous system, it is the preganglionic neuron which leaves the nervous system, travels to the ganglion where its synapses, with the postganglionic neuron, which would then go to the effector organ. There are other differences as well. So for example, the motor neurons of the SNS, they're large cells and they are myelinated and heavily myelinated. And I'm sorry, I'll get back to that. So here's just a depiction of the dorsal root ganglion, just for reference what a ganglion is. So in the spinal cord, clusters of soma here in the gray matter, that would be called a nucleus. All right, but here we have a bulge. What is in the bulge? That is the soma of the sensory neurons. But notice that it's located outside the central nervous system. It's outside the spinal cord, so it's called a ganglion. That's the dorsal root ganglion, and it has the soma of sensory neurons. And then once again, there are other differences. So the motor neurons of the SNS, they are myelinated and highly myelinated. The autonomic nervous system, you have different kinds of fibers. They are smaller. The preganglionic neurons have some myelin, not as much as what we see here. So I've read that as lightly myelinated fibers. And then the postganglionic fibers are unmyelinated. Now, this will then have an effect on the messages which get sent, because messages can travel at different speeds along axons, some 2 miles an hour, some almost 300 miles an hour. And two of the things that make the differences are the larger the fiber, the wider the fiber, the faster the message goes. So wide fibers carry electrical messages faster than narrow fibers. And myelin, those glial cells, if you recall, which then surround the axon and make a lipid protein mix called myelin, cause the action potential to jump from one node to another. Fat myelinated axons carry messages most quickly. So the messages of the autonomic nervous system with less myelin or no myelin on narrow fibers travels more slowly than those of the SNS. Finally, there are differences in the neurotransmitters which are used. So in the SNS, acetylcholine is released, and it always excites the skeletal muscle cell, causing it to go to action potential. So acetylcholine is an excitatory neurotransmitter released in the SNS. In the ANS, you can use acetylcholine or norepinephrine, and both of these could either be excitatory or inhibitory, as I'll explain. So there are more neurotransmitters and more varied effects in the autonomic nervous system. Once again, our central nervous system has this sensory division for information coming in, the motor division for skeletal muscle. Now, some of this control is voluntary, some is involuntary, as the extrapyramidal system with its nuclei deep in the cerebrum and the cerebellum and the brainstem would would have. But here we have more in the autonomic nervous system, the targets are more varied. Now, one little comment, because the idea of nerve cells innervating adipose, that's a little odd. So I'd like to, just explain that. When we think of how nerve cells send commands, in many cases, there is a synapse where this part of the nerve cell, the synaptic knob, touches another cell. And the second cell, let's say it's a skeletal muscle cell, is often specialized there. So there's a discrete structure called the synapse. So here's smooth muscle that the autonomic nervous system would send commands to. So there is a discrete synapse. But we really don't see that to the same degree in adipose and certainly not in immune tissue. Why is that? Well, because here, this is what we think of as a synapse. This synaptic knob is contacting a specific part of the skeletal muscle cell called the motor end plate. It's specialized for this synapse. However, in the autonomic nervous system, it can get more interesting because you can have the axon have a series of enlargements, varicosities, and they can release neurotransmitters along the length. It just doesn't have to be released from the very tip here. So that's different. A second difference is that in a tissue, this could move slightly so that its position could change. And there are cells of the body like white blood cells. They are always moving. Now, white blood cells, they make receptors for signals that the nerve cell is releasing. I mean, heck, if we want to get into it, the nerve cell is also making receptors for signals that the white blood cells are releasing. So they talk to each other, but interestingly, they can both move. The axon, not all that much. But the white blood cells could get a message and then leave and go act on that message. And so therefore, in the autonomic nervous system, there are a number of commands being given that would be harder for us to understand simply because, it's harder to study if both partners can move. And so in the autonomic nervous system, the targets are a bit more varied. Okay. As I will describe in just a little bit when we kind of get into the physiology of it, there are two branches of the autonomic nervous system, and thus kind of two modes for, say, the guts. If the somatic nervous system is sending commands to skeletal muscle, the autonomic nervous system, succinctly, sends commands to the guts, the cardiovascular system, the digestive system, the urinary system, the respiratory system, the reproductive system. And your guts kind of have two modes. Are you exercising or is it an emergency? That's what we call the fight or flight mode. I'll get into that. Or are you, not in an emergency or exercising? So are you in the rest and repose mode? The fight or flight mode is governed by the half, which we call the sympathetic division of the autonomic nervous system. All right. And so here you'll see, as I'll get into, things like the heart rate would increase, blood gets sent to muscles, etc. So that does, as opposed to the parasympathetic division, which will send commands to increase the activity of, say, the digestive system, the urinary system, the reproductive system, etc. So the autonomic nervous system has these two halves. How do they differ? Well, functionally, that's easier to talk about. What's the difference between an emergency versus not? But let's kind of talk anatomically for right now. The sympathetic division on the left is also called the thoracolumbar division because the thoracolumbar division has its preganglionic soma in the spinal cord in the thoracic and lumbar region. So remember that there are these two neurons. Where does the first neuron start? Well, for the fight or flight mode, the sympathetic division, the the preganglionic soma is in the thoracic and lumbar sections of the spinal cord. So it's also called the thoracolumbar division. In contrast, in the parasympathetic division, the preganglionic soma are in the brainstem and in the sacral division. So it's sometimes called the craniosacral division because the preganglionic soma are starting here and here. So there are neurons that start in the sacral division, and there are neurons which leave through a variety of cranial nerves. It should be stressed, however, there is one cranial nerve, the vagus, which carries most of these fibers. So the 10th cranial nerve starting in the vagus and then passing through the jugular foramen. This carries the vast majority, 80–90% of the parasympathetic division. So once again, it is in the cranial sacral division. So cranial, meaning that the preganglionic soma are starting, in the brain. And while other cranial nerves can carry these fibers, as can the sacral divisions, it is once again the vagus nerve which carries the vast majority of them. So that is one anatomical difference, but there are others. In the sympathetic division, the preganglionic soma, they are short, all right? It is the postganglionic, the preganglionic neurons, they are short. The postganglionic neurons, they have long axons. And so if they're short, if you look at where the ganglia are, you'll notice here that there are all these little white dots very close to the spinal cord. All right, so those are sympathetic ganglia because the preganglionic neurons are so short. They then reach these ganglia, which are all alongside the spinal cord. Now, there are a few other ganglia in the sympathetic division, which are not alongside the spinal cord like these here. Whereas in contrast, the preganglionic axons of the parasympathetic division. Notice that they don't reach here or here. Instead, they're going to the effector organ, which could be the lungs or the stomach or something. And their ganglia then is either near or inside the effector organ. All right. And so in the sympathetic division, the preganglionic neurons are short, and the ganglia are close to the central nervous system. The postganglionic neurons are then longer, traveling to the effector, whereas in the parasympathetic division, the opposite is true. Because of that, in the sympathetic division, let's say this one single preganglionic neuron, it could synapse with this neuron, which say goes over here to the heart, this neuron, which goes over here to the lungs, etc. It can go lots of places, all right? Because, starting from this point in the spinal cord, you could diverge a great deal. And so therefore, if this one neuron sends a message, which then gets relayed to lots of different structures, this allows a complex, coordinated response. So during an emergency, we see like the heart and the lungs and all of these body structures acting, together for a complex, coordinated response, in part coming from this degree of divergence, where one preganglionic axon can send lots of messages out. In the parasympathetic division, we don't see that. And so as a result, if this synapse is actually within the effector organ or next to it, one preganglionic neuron, synapses with an average of, say, 3 postganglionic neurons, allowing a limited specific response, which might, say, just affect the stomach, for example, but not nearby structures. So that is another anatomical a difference that we see between the sympathetic and the parasympathetic divisions. Now, the somatic nervous system releases acetylcholine. Acetylcholine binds to receptors on muscle, and this allows then ion channels to open. Ions come in and stimulate the skeletal muscle. So there are acetylcholine receptors here. In the autonomic nervous system, we see not only acetylcholine, but norepinephrine and epinephrine. There are differences, however, because, and I'll get into this again, the preganglionic neurons are always releasing acetylcholine, but norepinephrine is more of a sympathetic neurotransmitter, while for effector organs, acetylcholine is used by all of the postganglionic neurons of the parasympathetic division. One other difference, which I'm going to get into in much greater detail, is in the sympathetic division, there are neurons which go to the middle of the adrenal gland, what's called the adrenal medulla, and cause epinephrine and norepinephrine, which we also call adrenaline and noradrenaline, into the blood. And once they're in the blood, they act like hormones rather than neurotransmitters. All right, so I'll get into that now. So, the kidneys have two important glands which lie over them, one on each side, the adrenal glands. Now, when I teach in class, I tell a story, but let me just kind of make a more visual version of the story. So there is a museum in Connecticut, the dinosaur, the state museum, where there's footprints of Eubrontes, which is a Dilophosaurus-like dinosaur, which made these footprints. Wonderful, wonderful. So here's this. So let's imagine you go to this museum, and at one point there's a statue of the dinosaur that they think made these footprints. Let's say you didn't notice the statue, and then all of a sudden, out of the corner of your eye, you see it, and it startles you. All of a sudden there's this big thing that you didn't expect to be there, and you look and you are startled. Then you realize that it's just the statue in the museum, and that everything is good and it's kind of funny, and you wave at your friends, all good. I didn't mean to scream or whatever. Okay, so there you see the nervous system. Fast but brief. You whirled around, you saw things, you realized, what it was. The nervous system was acting. It acts very quickly, but the responses are brief. So if I want to do this, that's a nervous system response, fast but brief. If this individual, however, were to then measure their breathing and the heart rate, 2 minutes later, they might say, wow, I'm still startled with that, like my heart rate is still up. You already know that it was just a statue. Why is your heart rate, just starting, to now go up? And why will it stay elevated long after, you know that it was just a statue? And the answer is that in the sympathetic division, not only do we have neurons which go and touch their effectors. So for example, there are cardiac acceleratory nerves, which will actually go and touch the cardiac muscle of the heart, and like say the SA node, where it can increase heart rate. So in addition to that, the sympathetic division sends these nerves which go and touch the heart. There's also a neurotransmitter which is released into the blood, epinephrine and norepinephrine. And these neurotransmitters, it's going to take a while to get to the heart. So think about it. So if it goes into a vein in the adrenal gland, then the veins have to bring blood back to the right half of the heart. The right half of the heart has to send blood to the lungs. The lungs then have to send it back to the left half of the heart. The left half of the heart pumps it throughout the body. And now it says gets to this cardiac muscle. So it's not like this. See this? That's a nervous response. You can't get a hormone through the circulatory system to the heart that quickly. So this epinephrine is part of the stress response, but its effects will be slower to start. It's going to take a while to get to the heart, but then longer lasting, right? Even though now it was just a statue behind you, there's no reason to elevate your heart rate. Nevertheless, your heart rate is going to go up. So this is a unique aspect of the sympathetic division, which we don't see in the parasympathetic division. Epinephrine and norepinephrine, what we call adrenaline, noradrenaline, are released into the blood where they act as hormones. All right. So this adrenal gland has a cortex, and the cortex makes lots of interesting hormones, very important in the stress response, like the glucocorticoids, but that's part of a different lecture, and a middle, which we call the medulla. All right, so there's an outer cortex, and there is an inner medulla. The preganglionic axons from thoracic splanchnic nerves reach the adrenal medulla, where they synapse with cells that release epinephrine and norepinephrine. So then once again, it is here in the medulla that then epinephrine, which I'll abbreviate as E, and norepinephrine, NE, where they are released. But once again, they're going into the blood. So if a neuron releases norepinephrine to an effector cell like a muscle, that's a nervous response caused at a synapse. It's quick but brief. However, if this same signal is released into the bloodstream, it's not a neurotransmitter anymore. It is now acting as a hormone. As a result, that's why the response is slower to start and lasting longer. But it is an endocrine response, because now the nervous system is affecting non-innervated cells. So the sympathetic division can affect cells that it does not touch. All right, so that's impressive. And that's why this comes up again when we discuss the endocrine system and its hormones, because here norepinephrine and epinephrine are acting as hormones. Now, for a lot of students, what I just said is good enough. I'd like to add just a few more details here. This adrenal medulla is then unique in the body. Now, all of the nervous system is unique, but this area, just a little background, is when the nervous system forms in an embryo. It forms as first this plate, which then forms these folds, and the folds then fuse together and form the neural tube, which becomes the brain and spinal cord. Interestingly, however, notice the cells here in green. They are on the edges of the fold. So right where the fold comes together to form the neural tube, we have these cells, which we call neural crest cells. They are literally on the crest of the folds, which will form the neural tube. They are really important because it ends up, they make a lot of very important vertebrate-specific tissues of the head come from neural crest cells. Dorsal root ganglia come from neural crest cells, but so then do the ganglia then of the autonomic cells. So sympathetic, and so a lot of the neurons of the autonomic nervous system are going to differentiate from these neural crest cells, as will cells which are called Chromafin cells, which make the epinephrine and norepinephrine. So the cells which are going to release, so here you can see a dorsal root ganglion forming from those neural crest cells, which were outside the nervous system, the central nervous system. These Chromafin cells then are derived from the same embryonic precursors as the neurons of the autonomic, of the sympathetic neurons of the autonomic nervous system. They're called Chromafin cells because originally they were stained with chromium. They had an affinity for chromium, hence the name. It turns out that there are different kinds. So some are releasing norepinephrine while others convert norepinephrine into epinephrine. So they don't release both. Some cells produce one, some cells produce the other. The adrenal medulla, 80% of what it releases is epinephrine, 20% is norepinephrine. Now, when I talk about these, I'll just consider them to be about the same. Epinephrine and norepinephrine are functionally equivalent for the most part, but epinephrine is more powerful, and it has a greater effect on the dilation of respiratory passageways in the lungs. Norepinephrine is more significant, affecting vasoconstriction of blood vessels and thus affecting blood pressure. So I'll just put them together, epinephrine and norepinephrine, but the two are a little bit different. They are made from the amino acid tyrosine. So tyrosine has this ring. That's important because that's where the term catechol will come. And being an amino acid, it has an amino group. So if you eat tyrosine as an amino acid in your diet, some cells of your body then change it a little bit. They have enzymes which will, remove an H, add an, make it an OH, et cetera. And so here we turn tyrosine, which is an amino acid in your diet, into what we call dopamine, which is now a neurotransmitter. So this neurotransmitter dopamine has been derived from an amino acid. It can be called a catecholamine because it has this ring structure. It's a neurotransmitter which has a ring structure and an amino group. And so it's called a catechol, this part, amine. But then two other neurotransmitters can be derived from dopamine. And since dopamine is a catecholamine, then epinephrine and norepinephrine would likewise be catecholamines. So if you make this enzyme, the dopamine beta hydroxylase, that can change dopamine into norepinephrine, which a number of neurons actually release, as well as some Chromafin cells. But some Chromafin cells then make another enzyme, the phenylethylamine and methyltransferase, PNMT, and that will change, it adds this methyl group here, and that then changes norepinephrine into epinephrine or adrenaline. And so the Chromafin cells, because they make the right enzymes, they then can release epinephrine into the blood. And it turns out that we actually have Chromafin cells in a few other places. They don't have to release epinephrine into the blood, but they form from the same embryonic tissue. So the vagus nerve, the carotid, the bladder, the prostate, the liver, and also something known as the organ of Zucker-Kendall in either the abdominal aorta or in one of the branches. We also have Chromafin cells there. Okay. So that's a significant part of the sympathetic division, as is this. And forgive me, actually, I'm loading these into YouTube. This one I forgot to load, so this actually would have been a little bit better in this presentation 5 minutes ago, so I apologize for that. The sympathetic division can signal through epinephrine released as a hormone, but most of the signals are then once again going through those ganglia. The ganglia, which lie here, these white dots on either side of the spinal cord, they are called the sympathetic chain ganglia. So they're part of the sympathetic division, and they exist as a chain on either side of the vertebral column. If you want to call them paravertebral ganglia, you could as well. They're united by short nerves called sympathetic trunks or sympathetic chains, so that they are all connected. So it looks like a chain of beads on either side of the spinal cord. They can vary a bit in number. Here are the typical numbers here. And the way that it's structured is that the preganglionic soma is located in the spinal cord, and interestingly, in the lateral horns of the spinal cord. So for example, if you were to look at the spinal cord at the sacral level, you wouldn't see this red region, the lateral horns, that's found in the thoracic and lumbar region. So from the lateral horns of the spinal cord, we then have the axon of the preganglionic neuron, leave going through the ventral root, and then entering the spinal nerve. Now, if you recall, these neurons are myelinated. So the nerve then that comes out then looks white. So it's called the white ramus communicans because these neurons are myelinated, and the myelin, being part lipid, has a lighter color. So these fibers are in the spinal nerve, but they then leave the spinal nerve because remember, they can't get to the effector organ because in the autonomic nervous system, that's not how it works. It's the second neuron, the postganglionic neuron, that gets to the effector structure. So these then are going to leave the spinal nerve and go to the sympathetic chain ganglia, where they then synapse with the post-ganglionic neuron. So the preganglionic neuron, its soma, is in the central nervous system, in the spinal cord. Its axon comes out here and ends. This is where it synapses with the postganglionic neuron. Now, there are some variations. So for example, it's typical that this axon would synapse with in a ganglion that is at the same level. So here we'll see it at the same level. It came out there and it goes there. Here then is the postganglionic neuron. And it's now going to send its axon back to the nerve so that it can go to the effector organ. Remember that the postganglionic axons are unmyelinated, so it's what's called the gray ramus communicans, because without the myelin, it has a duller color. So the white ramus has the preganglionic axons, which are myelinated, and then after the synapse in the ganglion, the gray ramus then goes back to the spinal nerve with the unmyelinated fibers. There is some variation. So this preganglionic neuron then doesn't synapse in the ganglion, which is next to it at the same level, but actually it might go to one sympathetic chain ganglion above or below and synapse there. So that's a possibility. And then some of these white fibers actually don't synapse in the sympathetic chains on either side of the spinal cord. They will go elsewhere to another ganglion to synapse there. Now, some of them might run, say, now underneath the spinal cord, all right, ventral to it, and now form what's called a splanchnic nerve. So you'll see this one, it did not synapse in the ganglion. It went straight through. It's now going to go alongside the ventral side of the spinal cord as a splanchnic nerve, and they can get really complicated. They can interweave. So some go this way, they swap fibers, they form this, net called a plexus. So there are celiac and mesenteric plexuses, for example, where these splanchnic nerves can interweave, and they can even be, complex because now even, other parasympathetic postganglionic neurons can fuse. So it can be confusing. So, just one last thing, but beyond a certain point, the anatomy of the autonomic nerves gets extremely complex. One last thing is in the head, that's different. So we do send autonomic commands to the head. So, we dilate our pupils, we make saliva, etc. And so preganglionic axons leave the brainstem and travel through several cranial nerves, the ocular motor, the facial, and the glossopharyngeal nerves. They then send their preganglionic fibers to ganglia in the head region. And then many of the postganglionic neurons then go through the trigeminal nerve. All right, so there are a couple of cranial nerves where the preganglionic fibers leave, and then the postganglionic fibers then have different routes, some through the trigeminal nerve. But once again, it is the vagus nerve, cranial nerve 10, which carries the majority of the parasympathetic fibers of the body. Okay. Now, the autonomic nervous system, I have found in my teaching, is something that students don't like the anatomy of, but the physiology is kind of fun, and we're almost on the anatomy. And so a couple final points in the anatomy of the autonomic nervous system. One is the difference between cholinergic and adrenergic. Cholinergic refers to the release of acetylcholine, whereas adrenergic refers to norepinephrine and epinephrine. So here you see something different. You see serotonin, and obviously there are neurons which have to make serotonin, right? If that's going to be the neurotransmitter of choice, they then release serotonin at a synapse, all right? And then serotonin binds to receptors, okay? And so this cell made the neurotransmitter. This cell made the receptors for the neurotransmitter. And then in the same way, when we look at, say, acetylcholine and norepinephrine, there will be some cells which make it and other cells which make the receptors for it. So a neuron which makes acetylcholine is called a cholinergic neuron. A neuron which makes norepinephrine is called an adrenergic neuron. All right. So as stated, I'm sorry, I'll say it in a second. All of the preganglionic neurons release acetylcholine. So in both the sympathetic division and the parasympathetic division, the preganglionic neurons release acetylcholine, so they are cholinergic. Whereas in the sympathetic division, most of the postganglionic neurons are adrenergic, releasing norepinephrine, while in the parasympathetic division, the postganglionic neurons are also cholinergic. So neurons are called cholinergic. They release acetylcholine. This neuron's releasing acetylcholine. It is cholinergic. And in the sympathetic division, that applies to the preganglionic neurons. And in the parasympathetic division, it applies to the preganglionic neurons. In contrast, however, the postganglionic neurons can vary. So in the parasympathetic division, the postganglionic neurons also release acetylcholine and are cholinergic. However, the majority of the sympathetic postganglionic cells are releasing norepinephrine and are called adrenergic. So just a definition, cholinergic versus adrenergic. However, then we can also then ask, how do we respond to receptors? Well, a receptor is cholinergic if it responds to acetylcholine. A receptor is adrenergic if it responds to epinephrine or norepinephrine. So this gets a little more complex. So once again, neurons are cholinergic or adrenergic if they release the neurotransmitter. But obviously, this neuron releases norepinephrine, noradrenaline. So it's adrenergic. But obviously, if this is the signal which is coming to it, it has to make on its soma and its dendrites receptors for acetylcholine. So even though it's an adrenergic neuron, it is making receptors for acetylcholine here, all right, but it's releasing norepinephrine here. The effector, whatever this is, so if this is, say, going to smooth muscle, the smooth muscle then has to make the receptors for norepinephrine. This cell will make cholinergic receptors here, and then the smooth muscle that it will touch will make adrenergic receptors. So notice that the receptors for acetylcholine have to be wherever acetylcholine is taking an effect. So that would be here, here, here. Those are the cholinergic receptors. And then adrenergic receptors would obviously be on the effectors. So the sympathetic division, once again, that's how most of the neurons work, where all of the preganglionic neurons release acetylcholine, see all three of them. Most of the postganglionic neurons are releasing norepinephrine. In some cases, in the adrenal gland, those Chromafin cells, they're releasing acetylcholine. My fault. The Chromafin cells are releasing epinephrine and norepinephrine here. All right. So that's different from what typically happens, but this is just in the adrenal glands. And in a minority of the postganglionic neurons, such as those going to sweat glands, they would actually release acetylcholine as well. So in the sympathetic division, notice that the second stage can vary. I don't want to get into the argument. There is an argument over these chromaffin cells. Are they actually the postganglionic neurons which are modified? Are they similar to the postganglionic neurons being derived from the same type of embryonic cells but distinct? So if then that's the case, are there postganglionic neurons which just don't have soma, or just don't have dendrites? In fact, what are they doing? If there are no postganglionic neurons, then this actually isn't a ganglion, so this can't be called a preganglionic neuron. So we could argue a little bit over the embryonic lineage of this and how that affects our naming, but let's not argue. Okay, so that was one definition on cholinergic versus adrenergic. And then another couple definitions which are extremely important in pharmacology is on those receptors. So if there are receptors for acetylcholine, the cholinergic receptors, and receptors for epinephrine and norepinephrine, the adrenergic receptors. There are luckily multiple kinds of them. Okay. Now, just a quick analogy. If you were to say it's cold and it's snowy, all right, is that good news or bad news? Well, if you have to drive through this, that's bad news. All right. So if you said it's cold and snowy today, the weather, some people would be saddened by that. That's bad news. I have to drive through this. But then other people might realize, hey, we get to, play, a variety of winter sports. That's good news. So notice that the same news, it's snowy, can then either be good or bad news, depending on the receiver and how they respond to it. All right, so it's snowing. That's not automatically good or bad. It depends on how you respond to it. Same thing here. This neurotransmitter, is it excitatory? Will it excite the second cell and get it closer to action potential? Or is it inhibitory? Well, that's actually not determined by the neurotransmitter, but by how this cell responds to it. One cell might respond to, say, acetylcholine and say, hey, that excites me, I'm going to go to action potential. While a different cell may then get inhibited. So acetylcholine, for example, is the message which tells skeletal muscle to contract. But acetylcholine, the same message, will actually inhibit cardiac muscle and decrease heart rate. So acetylcholine will tell, excite skeletal muscle, but actually inhibit cardiac muscle in the heart. So when we want to slow down heart rate, the vagus nerve releases acetylcholine. Or I'm sorry, it's postganglionic cells after synapse. So therefore, these receptors then matter. And so we then look at the cholinergic receptors, which respond to acetylcholine, and we realize that there are different kinds of them. Some are called nicotinic if they respond to nicotine. Nicotine can stimulate these. Others are called muscarinic if they respond to the mushroom poison muscarine. And the receptors for adrenaline and noradrenaline, they vary. They can be called alpha and beta. And then there are subclasses of each, alpha 1, alpha 2, beta 1, 2, and 3. And the muscarinic receptors, they have subclasses as well. All right. And so when you ask, what is the effect of acetylcholine? What is the effect of norepinephrine? Well, it depends on what receptors the effector cell is making. So both of these receptors are called cholinergic receptors because they're responding to acetylcholine, but they're distinct. All right. So there's a difference between the nicotinic receptors and the muscarinic receptors. The nicotinic receptors are those used by all of the postganglionic neurons which, on their soma and dendrites. All right. Skeletal muscle cells will use them as well. They are used by the Chromafin cells in the adrenal medulla. And when acetylcholine binds here, it excites this the cell. So these are always excitatory. In contrast, the muscarinic receptors are found on the effectors of parasympathetic organs, and in those eccrine sweat glands, which I said that's the exception where they also are part of the sympathetic division, but they are responding to acetylcholine. And here there are different kinds of muscarinic receptors. Some will excite the effector cells, some will inhibit the effector cells. So acetylcholine can cause a smooth muscle to depolarize and get closer to action potential, but have the opposite effect on cardiac muscle. And then in the adrenergic receptors, the same thing applies. So we have different receptors responding to norepinephrine and epinephrine in blood vessels, the pupil of the eye and sphincter muscles, where it's alpha-1, in the pancreas affecting insulin release, where it's alpha-2, in the heart, the kidneys and adipose, mainly the heart, where it's beta-1, the lungs, blood vessels of the heart, liver, skeletal muscle, beta-2, and adipose, where it's beta-3. So there are different kinds of receptors. Receptors for acetylcholine are cholinergic are nicotinic or muscarinic, and the receptors for adrenaline and noradrenaline are alpha or beta. Who cares? All right. Well, just big picture, and this is where we're about to end the anatomy portion. But pharmacology classes, I mean, this is one of a central concept. What do people seek medication for? Well, obviously, a lot of people are on heart medication to lower blood pressure or to affect, heart rate, etc. A lot of people take medications which affect their lungs. So this might affect, for asthma. But then also, all of these, whether it be the secretion of glands, the digestive system, the urinary system, the reproductive system, let's just say the guts. Lots of people take medications which affect the guts. If there was only one kind of receptor, well, that would be bad, because then a medication which affects that one receptor would not just say affect the heart the way you wanted it to if you have high blood pressure, but would affect the lungs and the urinary system, the reproductive system and the digestive system, etc. But because there are all of these different kinds of receptors that are found in different organs, then one medication could affect one receptor without affecting all of them. So you could treat asthma without affecting high blood pressure, or affect high blood pressure without treating asthma or glaucoma or the ease of urination or congestion, etc. And so, there are selective, say beta blockers, non-selective beta blockers. So a very important concept in pharmacology is once you have this great diversity in receptors, then you can have a great diversity of medications which affect some receptors and not others. Two final concepts for this portion. One is the idea of dual innervation. So the heart does not belong to the sympathetic or parasympathetic divisions. The sympathetic division sends nerves to the heart to speed up heart rate, and can speed up heart rate through epinephrine. And the parasympathetic division sends a branch of the vagus to the heart to slow heart rate down. So the heart is dually innervated. It has dual innervation by both the sympathetic and the parasympathetic divisions. So does the stomach. We can digest food more, increase gastric motility and secretion when we're at rest, but we can stop it when there is an emergency. So both the sympathetic and parasympathetic divisions send information to the stomach and to the lungs. We can breathe faster, more deeply, more rapidly with bronchodilation when it's an emergency, or we can breathe less and have bronchoconstriction when we are at rest. So most organs of the body receive both sympathetic and parasympathetic input. That's what we mean by dual innervation. There are some exceptions. So for example, the eccrine sweat glands, which release that watery sweat to cool us down, they receive sympathetic input, but not parasympathetic input. So there are some exceptions, such as sweat glands. Final point is lots of regions of the brain and spinal cord are involved in the autonomic nervous system, and lots of parts of the brain then have input into it, such as nuclei in the reticular formation. Obviously, the autonomic nervous system involves heart rate and breathing, and it's the medulla oblongata, which has a cardiovascular center, a respiratory cardiovascular center, a respiratory center, etc. So lots of brain regions are involved. That being said, the hypothalamus is particularly important as a central organizing center. So even though, once again, lots of parts are involved in the autonomic nervous system's control, if we were to look at the hypothalamus, it is incredibly complex, so complex that look at all of these nuclei in the hypothalamus that we identify. A number of these then are involved in coordinating the autonomic nervous system. So if you were to pick a place in the central nervous system that is the control center for the autonomic nervous system, it would be the hypothalamus. Okay, so this is extremely important. And in anatomy, classes, very often it's like the anatomy 1 students, which are studying the brain, and the autonomic nervous students. But anatomy and physiology part 2, that's where you talk about the heart, the lungs, the digestive, urinary, reproductive systems. And so the autonomic nervous system, which is then the boss of, say, the guts, that's extremely important in anatomy and physiology too. This ends the lecture on the anatomy of the autonomic nervous system, and the next lecture will go through the fight and flight response and the rest and repose response.